So what kind of operating systems we will be working on when we are dealing with the variant calling process? So we need a Linux operating system, or if you're on Windows operating system, you just need WSL terminal for your Windows operating system. So in the previous section, I think you should have got the WSL introduction, how to install and how to make Windows subsystem for Linux on your operating system as well. If you're on Linux, you don't have to do any of that. Just move on to the process that we are going to do now. So what are the softwares and setup that we need for variant calling process? So list of system and softwares that are required. So the software that we need, first one is wget or curl system integrated into our terminal. So we need to install a wget or a command line process for our interfaces. For example, we need wget or curl command line tool for accessing the resources on the internet. So they have slightly different command line interfaces. For example, curl is for the, I think, bash operating system. We also need that in by using the curl you can do all the web scraping on the iMac, I think, so Mac operating system as well. So second tool that we need is called fastqc. So you can install fastqc using the apt system, which is also sudo apt install whatever on the command line, because fastqc is the standard package archive, just as same for the porchop. So we will be using porchop for the quality trimming. So what is quality trimming? I'm gonna explain all that as well. So the main tools that we're gonna use are the Samtools. Samtools are also available on the apt system, sudo apt install Samtools, and bcftools, one of the main tools that we are going to use. Also available on the Ubuntu operating system, so you can use your bcftools sudo apt install. So what will be the basic workflow that we need for the NGS calling analysis or variant calling analysis? So we need raw reads, then we need the quality control, then we need the quality trimming of data. After quality trimming, we need the alignment with the reference genome. For alignment purpose, we need reference annotation and a reference genome which contain the fast try sequences. Then we have to compute the depth and coverage computation. After that, we are going to call variants. So that was the basic introduction of the workflow. Now understanding the data set that we are going to use. Which kind of data is used for the variant calling analysis and what is hypermutability in that process. So the sequencing data from the Illumina sequencer. So Illumina is a company that does all the sequencing analysis, which uses the flow cell, which are divided into lanes, which is also going to divided into tiles. The FASTQ file we will use later has the information on the lane tiles position. Keep that in mind. We are also going to see a FASTQ file. Then I'm gonna explain that where are the tiles, lanes, and flow cells information. It is useful to familiar with the general organization of the data. As shown, the exact number and the layout of the tile, it process a lot of data for itself. So here are the high-sec flow 8 cell lanes. You can see one lane contain 120 tiles, and one tile contain up to 5 billion read per flow cell on high-sec 4000. High-sec 4000 is the type of analysis that we do in NGS. So what is the metadata that we are using? So we will be working on the 3 sample events from era 3 strains of this experiment, one from the 5000 generation and one from the 15,000 generation, and one from the 50,000 generation. The population changed subsequently during the course of the experiment. So we will be exploring how will the mutation evolution of SIT mutant and hypermutability with our variant calling workflow. So that was the hypermutability, how to check that mutations in your hyperdata. So now that we have discussed the data, the basic workflow, and all that stuff, First one, we will be moving on to getting the raw reads into our system, how to get all the raw reads that we need, how to retrieve the raw reads, how to use the SRA toolkit. So that will be analyzed in the next, next lecture. Yes, in the next lecture, we are going to retrieve the reads. So that was it for this lecture.